Assalamu alaikum students. Today we are going to start the fifth chapter of biology, that is cell cycle, in which we will talk about all the different phases that takes place in the whole cell cycle and the process of mitosis. The first thing that what actually this cell cycle is, it is actually a series of events that is a scientist actually identified that are repeated cycles of events in the life cycle of cells takes place. Cells actually consist of the two major phases, which is called as the interphase and mitotic phase. The cell cycle of events, the whole cell cycle that takes place, is called as a cell cycle. Next thing is about the cell division, how the cell divides, what happens in the cell division. Every hour, about 1 billion cells die and 1 billion cells are made in our body. Part of the cell cycle includes making new cells in the process, and it is called as the cell division. That means that through the cell cycle, one cell is dividing and making number of different cells. It is called as cell division. As you can see in this figure, that from a single cell, number of other cells are also originating. This process is called as cell division. Then the cell cycle takes place. Cell cycle consists of number of different phases. The first one you can see in the figure is called as the growth phase, and it is G1 phase. After that, the S phase starts, in which the DNA replication occurs. Then G2 phase starts. In the G2 phase, there is the preparation of mitosis. After that, the M phase starts. M phase is called as the mitotic phase. In the process of M phase, there are four different steps during the M phase. The first one is the prophase, metaphase, anaphase, and telophase. After that, the cytokinesis occur, and this whole process leads to the cell division. As we say that in the previous slide, it is divided into two parts: the interphase and mitotic phase. So these G1, S phase, and G2 phase are called as the interphase, while rest of these phases are called as the mitotic phase or cell division. Then the cell cycle has four phases: G1 phase, S phase, G2 phase, and M phase. And the first three are called as the interphase. Then what is interphase? Interphase actually composed of the G1, S, and G2 phase, where cell spends most of its time. Is mostly you can see in the figure that G1 phase is taking about 5 hours, S phase is taking about 8 hours, and then the 4 hours in the G2 phase. While it will take just a few hours will be taken during the M phase. So the 90% of the time will be spent in the interphase, often called as the resting phase. But cell is not at rest during that time. The division process is not taking place in these three phases. That is why it is called as the resting phase. And the cell is not dividing, but it is active. So interphase is like the resting phase. You are on the rest, you are not doing anything. That means the cells are not dividing. In chromosomes, I will tell you the few of the terms that we can use in this chapter. The first one is the chromosomes. Chromosomes are stored in the nucleus, and every cell that contains a nucleus will have the chromosomes. Here is the figure of the chromosome, because chromosomes look like this. Chromosomes contain DNA, you can see in this figure. Then DNA. DNA is actually the genetic material, the genetic code through which the information is stored. So genetic information is actually stored on chromosomes. The code information on chromosome is called as DNA. This is not just a helical spring-like structure. It is actually the coding. There are a number of codes present on it, and this code is making the DNA. This DNA stands for DNA ribonucleic acid, and this code is telling us that some of the information is there, we come to know that what is the hair color, what is the eye color, what are different characters of the body can be stored in this DNA. What is the structure of chromosomes? It consists of two things: chromatin, and chromatin is what the unwound form of the chromosome. Like if it is unwound, if it is open, that is called as chromatin. The other form is condensed, which is the tightly wounded part, like this one, right? It is making the coil, and it is saying that it is the condensed form of the DNA. This one is the chromatin. Like only the thread-like structures is called as the chromatin. Then, what is chromatin? Chromosomes unwind when the cell is not dividing. Cells divide नहीं हो रहे, तो वो unwind हुए हैं, तो हमने क्या कहा? That is the chromatin. So unwound chromosomes are available to be read by the cell's machinery. Cell's machinery read it, and it will form what? This cell machinery is helping to read the chromatins. For example, the open book, the DNA is accessible. Thus, the cell can carry out the instruction coded in it. Means if it is unbounded, it is like the open book. The DNA is accessible for to be read. So thus the cells can carry out the instructions coded in it. All the instructions that get coded in it can be easily read. So they are not visible under the microscope. Then condensed chromosome. Condensation occurs at the beginning of the cell division. Condensed chromosomes are unavailable to be read like a closed book. So visible under the microscope. These can be visible under the microscope. But if it is unbounded, then it can't be easily. It can be shown as a red spot, but not like there are some chromosomes present here. Okay, coming towards the phases of mitosis, the G1 phase. G1 phase is the first growth phase. It is actually the period of rapid growth. In the G1 phase, the new proteins and organelles are produced. Then the chromosomes are unwounded, means the chromatins are present. It also prepares for the DNA synthesis. That means it is preparing cell for the S phase. The next phase is S phase. What is actually the S phase? In case of the S phase, the cells make a copy of its entire set of chromosomes. It will make the whole copy, and why is it necessary? Because it is making the copies and it is helping the DNA to divide, right? And what would the chromosomes look like now? You can see that the chromosomes, why the chromosome is duplicated now, right? Chromosomes that have a duplicated will have two sister chromatids attached to the same centromere. So sister chromatids have the identical genetic information. उनके अंदर जो genetic information stored होती है, that is identical. So after that, the next question is, what would be the chromosomes look like now? The chromosomes remain the same. Its look will not change. After that, the next phase starts, that is the G2 phase. We know that it is the phase for the preparation of mitosis. So cell grows larger in size in preparation for cell division. Then it produces organelles and structure for the cell. And for example, the centrioles and nucleolus, they all started duplicating. You can see in the exam figure that there are two nucleolus are present here. So that means it duplicated. And then G2 phase, gap 2 phase is actually the shortest part of the interphase. Then the next phase will start, that is about the G0 phase or G0 phase. In this phase, the cell leaves the cell cycle, and it can be temporary or permanent, but not necessarily dead. So, for example, we can say the neurons are present according to it. Then next phase is M phase, that is the mitotic phase. We come towards this, where actually the cell division occurs. So all of the cell's energy is devoted to the process of cell division. Whole cells, the cell needs a lot of energy in this process. So M phase is divided into the two processes, mitosis and cytokinesis. Mitosis contains prophase, metaphase, anaphase, and telophase. You can remember it by remembering this word PMAT. Through the PMAT, that means prophase, metaphase, anaphase, and telophase. What happened in the process of prophase? That chromosomes condenses and no longer chromatin. It gets condensed now and become visible. Then nuclear envelope disappears. Centrioles move towards the pole of the cell. Like there are two different poles on each side. It moves towards there. Then spindle fibers begin to extend from the poles. After that, there are in this image you can say the chromosomes are condenses. nuclear envelope is disappearing. There is no nuclear envelope here. So it is actually the late prophase and it is the early prophase. In the start it looked like this, but later on it becomes like this one. In the case of chromosomes, what happened? That nuclear envelope disappears. Here there is no nuclear envelope. Then centrioles moving towards the poles. Centrioles come towards the poles and the spindle fibers will form. These long structures are actually the spindle fibers. After that, the metaphase starts. Chromosomes line up along the equator. Here's the cell and all the chromosomes are lined up in the equator, that is center of the cell. Then spindle fibers attach to the centromeres of each chromosome. From here the cell starts emerging and you can see this is the actual figure. The metaphase, in the case of metaphase, what happened? The chromosomes are arranged in the center. After that, what happened? From here the spindle fibers originated, and from these spindle fibers it gets attached to the centromere, that is central part of the chromosome. It gets attached to it and started pulling it towards its sides. So in the case of the third phase, that is anaphase, spindle fibers shorten, pulling chromosomes towards the opposite sides. Spindle fibers become shortened and the chromosomes are moving towards towards each pole. So sister chromatids separate in centromere and move towards the poles. Here is the actual example that chromosomes are moving to the poles. Then last one is telophase. Telophase is what? Chromosomes uncoil and become invisible. Nuclear envelope reappears. Spindle fibers disappears. It occurs simultaneously with cytokinesis, and daughter cells have identical genetic information. All that means that this one and this one, both the daughter cells have the identical genetic information because these chromosomes are making that this chromosome and this chromosome is making a separate cell. Then here is actually the chromosomes uncoiling and nuclear envelope reforming. What is the cytokinesis? That is separation of the cell and cell contents. Means the cytoplasm, organelles, cell membrane, everything is separating. You can see that it comes towards the different poles. Chromosomes are coming towards different sides. Nuclear envelopes appear and they are separating the two parts and it is making a new nucleus. This process is called as the cytokinesis. You can see that does not have to be an unequal division. Daughter cells can be of different sizes, have different cellular content, but nuclear content is the same. Like budding, right? You can see the example. This is the bud is forming, this is the parent one, and in case of the budding it also happens that it contains the necessary content from the parent, but it is obviously not the complete content but the part of it. Here is the cytokinesis in animal cells. It is actually a punching of the cell membrane and forming a furrow, and this furrow is helping to divide the one cell into two parts. You can see this, the nucleus is separated and the cell is separating from each other. This is actually a fluorescence we see this figure from the fluorescence microscopy. And this green part is showing the microtubules, the blue part is showing the chromosome, and the orange is actually the mitochondria. Then cytokinesis in plant cells. It is the formation of the cell plate. In the plant cell, the cell plate will form. You can see that here and here the two vacuoles are present, the nucleus is separated, and then the cell plate is formed here and two cells are separated from each other. Here is the other figure of the cytokinesis in plants. In telophase and cytokinesis, they look like same. The telophase and cytokinesis are also same at this point, and at the interphase they are also seen. That, like for example, we can say that in the nucleus the part of the nucleolus separated, while in case of the cytokinesis the part of the cell separated along with all the necessary components. Then what is the outcome of mitosis? Two daughter cells, each containing identical genetic information, and daughter cells have the same number of chromosomes as the original parent. So here is the overview of the cell division. That is G2 phase, centrosome, chromosomes, chromatin, then prophase occurs, metaphase occurs, anaphase, and then finally the telophase. And here is the fluorescence microscopy. That is that figure of the chromosomes. In the next one, we'll talk about the significance of mitosis. Actually, significance of mitosis is development and growth, and it consists of, like, for example, how the cell can divide, and it will help in the growth of the plant, and growth of the animal as well, growth of the human as well. Then the cell replacement, for example, one part of the cell is divided or separated from the body, like it all again reappeared. This is the cell replacement. Then, for example, the red blood cells, the new red blood cells will form if the one will die. So the regeneration, in case of the starfish, if one arm is gutted, then the other will form at the same place. The lost arm will form there. This is the regeneration. And then the asexual reproduction happens. In the case of asexual reproduction, the budding will occur, as I showed you in this figure. So in this one. So after that, there were some errors in the process of mitosis, that if the cells continue to divide, it may form the tumors. It can be the benign tumor, that is the minor one, or the malignant tumors, that is the cancerous tumor. So the excess mitosis is not helpful. It can be controlled, that is, if it is more, then it is actually the abnormal cell division. So this is all about the mitosis. In the next lecture, we will talk about the meiosis.